So for these three different regions, as you can see, the observed OTUs, which represent the diversity of the bacteria presented in the fecal samples, and the x-axis is the subject's age. You will find in the early life, so the diversity of the bacteria increased, especially from 0 to 3 years old. And this is the same trend for all the three different populations. There is a long-term interactions between the bacteria and the gut immune systems. As you can see in the upper part of this figure, this shows like in the prenatal stage, the colon, there is no bacteria colonized. This is like germ-free colon. And in the bottom, this is the postnatal colon. Let's see. So first, if we check this left figure, this is homeostasis status. The bacteria are mostly commensal bacteria. They are beneficial. So they maintain a structure here which will prevent the pathogenic bacteria to come in and colonize into the colon. And they can also train the immune system to produce like mature B cells and also produce the cytokines to prevent the pro-inflammation cytokines later in the life. However, if we check this right figure, this is dysbiosis status. You can see a lot of pathogenic bacteria colonize and compete with the commensal bacteria. They can also cause the tissue damage and cause skewed immune responses. So the idea is this bacteria, this commensal bacteria, really play a beneficial role in the beginning of life to train and fine-tune your immune systems, which will help you to avoid developing a lot of autoimmune diseases and allergies, asthmas. However, I should say that there are a lot of debates here, like in the clinical stage, whether this is a germ-free environment. More and more evidence shows actually there are some bacteria colonized before birth. And another pathways how the prenatal environment can cause the difference in the postnatal microbiome. Maybe because like the gut environment will change during the fetal status. So the environment may favor or help some bacteria to colonize. So it's like they have a selection pressure for some kind of bacteria. And also after birth, the mother's bacteria can transfer to the infant. So if the mother experienced some adverse exposure during their microbiome may be skewed and may be disturbed, and this disturbed microbiome can be transferred to the baby. So this is a model to show that from the conception of the baby, the mother and the fetal may have some microbiome transfer. And during birth, this is a colonization of the environmental bacteria starts. After that, we have a very critical window for the interaction of the microbiome and the immune system. And after that, in the early childhood, we have a microbiome maturation. So then this microbiome may influence the disease in adolescence and adulthood. So that's why the early infancy some experience can cause some disease like adulthood. So we are familiar with the hygiene hypothesis. So this is a working model very similar to the hygiene hypothesis, as you can see here. If the mother experiences more farm exposure during the pregnancy and during the infant contact with multiple farm animals and they consume farm meal, they will have a more adaptive immunity and the rate of asthma will decrease. So this really highlights the important role of microbiome in the early. And a lot of studies also shown that the maternal prenatal adverse exposures increase the risk for asthma. As you can see here, all the black bars are the children um, born to the mothers who experienced some stressful life events during pregnancy. And the white bars are mothers who did not experience such events. This is the prevalence of the wheezing or asthma or other allergy conditions in the children. As you can see, if the children were born to mothers who experienced these stressful life events during pregnancy, they have high risk of developing allergy and asthma later in life. So why we have this? Although there are many different hypotheses here, including microbiome as one of the mechanisms, we didn't have much study to characterize the microbiome of the baby who were exposed to some prenatally exposed to some events, adverse events. So this is the um, like United States report showing that the disasters are increased in the past 20 years from 2000 and 2019. As you can see here, a lot of disasters, like the number of disasters nearly doubled the number between 1980 to 1990. And most of them are related to extreme weather events as a result of the global warming. As you know, global warming have a very profound impact on the weather patterns. It's not only related to the weather increases, it's also related to weather decreases. For example, the current, the Texas winter storm. So we are expecting more frequent and natural disasters due to climate change. So it's very urgent to really understand how these kind of disasters expose, what's the impact of this prenatal maternal exposure to the disaster on the offspring. So we're using the Hurricane Maria as a model to do this study. So you know much more than me. So the Hurricane Maria struck Puerto Rico on September 20, 2017. Because of delayed response, a lot of regions experienced a long-term short of critical life resources, uh, loss of electricity, and I think a lot of like physical and mental disasters to the residents of Hurricane, to the Puerto Rican people. And the expected death toll is ranging from 3,000 to 5,000. And it's very unfortunate for such kind of like devastating disaster. But it's fortunate that the researchers on the island promote research, and many good studies come out, and it really contributes to our understanding of the big disasters on human health. And the researchers are very open to collaboration. Here, I want to thank my collaborators, major collaborators, Dr. Nicholas Rosario, Dr. Mita Acevedo, and Dr. David the Angel City. So they are all in the San Juan City Hospital, and we formed a collaborative team very soon in the beginning of 2019. We started to do this cohort study. So this study named Hurricane as the origin of later outbreak microbiome OLA study. This is also my first Spanish words I learned from the Dora book by with my daughter. So in this study, we have recruited infants who were exposed in utero to Hurricane Maria, and the recruit period is from April 2018 to August 2018. we totally have 29 recruited. Although this is a small number, but we really appreciated all the families because the mothers were still overwhelmed with difficulty due to malaria. And we also have a control cohort, which we recruited the infants 1 year later, the same period. So all the infants were conceived at least 5 months after hurricane Maria to avoid any adverse exposures to the mother. And we collected the nasal microbiome, the nasal sample, and stool. And you can see here, this is the PC plot, which plotted the similarity of each sample. So all the green dots are the gut samples, and all the yellow dots are the nasal samples. We totally have 63 nasal samples, 63 gut samples. And their composition are very different. As you can see, there are 2 clusters here. And I also want to note that all the babies are full-term, healthy, vaginally delivered. This is to avoid any confoundings in the microbiome study. So first of all, we compare whether there are differences in the composition of these 2 groups. The red dots are the exposure and the gray dots are the control group. Yes, we find they are different in the composition. So then we want to check what difference are they. And as you can see here, this is like 2 clusters. If we use the most abundant 10 genera to do the cluster, we can identify 4. And you may notice that for these 2 clusters, most of the subjects are from the exposure group. This is the bar. This bar shows the red is from exposure group. The black is from the control group. Each column is one. Each row is 1 bacterial genus. So you can see here. So totally, only 1 of the subjects are from the control group, but 14 out of 29 subjects in the exposure groups are within these 2 clusters. So one of the clusters are dominated by the bifidobacteria. So you can see the redness means the abundance are high, and if the color is yellow or almost no color, that means the abundance is very low. So this cluster is extremely dominant by the bifidobacteria. And on the other hand, for this cluster, you can see there's almost no bifidobacteria at all. We know that bifidobacterium and Bacteroidetes, these first 2 are the common bacteria in the infant microbiome. They are usually like the structure to maintain the structure of the infant microbiome. So for this group, they lost these 2 common bacteria. And for this one, it's predominantly by one of these bacteria and not the other one. So it's a little worrisome why these 2 are very different than these 2 seems very healthy clusters. We also checked using a different way. As you can see, this is the cluster with no Bacteroides or bifidobacterium. And this is a cluster only dominated by the bifidobacterium. For these 2, only 1 subject from the control group. All the remainings are from the exposure group. But for the other 2 clusters, they are like evenly distributed in the control of the exposure group. If we do the PC plot, as you can see here, the 2 skewed clusters are here, the yellow one and the green one. So we believe this is actually the reason why the exposure group and the control group are different. It's really because of these two clusters. So now the question we want to ask is whether, as you can see here, for this dominated by bifidobacterium cluster, whether there are some species which are very special in this cluster, which cause no colonization of other bacteria. So we did a metagenomic sequencing, which is different from 16S, because 16S sequencing usually they can only give you the genus level information. If you want to go down to the species level, you need to do the metagenomic sequencing, which shows all the different bacterial species. As you can see here, this is a bifidobacterium cluster. We didn't see any like very different bacterial species here, but we found that bifidum and Breve, these two species are increased in this group compared to the other. So this one is the cluster there's almost no bifidobacterium, but very, very little. But if we compare this as like to 100%, we can see there are about 3 different species within this group. And for one of these, this is like Shiwa, no heads. So these species are a little like unique within this group. But after all, this is the cluster which has almost no bifidobacteria, very less bifidobacteria. But this study shows that there is no very unique bacterial species within this cluster for the bifidobacterium. We need to do more research on why these clusters are so dominated by the bifidobacteria. So this is to identify the richness difference of these 4 clusters. As you can see here, the bifidobacterium, so this cluster have a significantly less richness of bacteria. We think this is because they are dominated by just one genus, the bifidobacterium. So therefore, there's no much space for other bacteria to colonize. And for this group, we didn't see a significant difference compared to other groups regarding the richness or very close to the alpha diversity of the microbiome. And then we did very similar studies on the nasal microbiome. As you can see here, for the nasal microbiome, if we use the top 10 abundant bacterial genus to do the cluster, we have totally 3 clusters. They are dominated by Corynebacterium, Dolosy granulum, or by some other pathogenic bacterium, as you can see here, the Moraxilla or Salmonella and Streptococcus or Haemophilus here. But we didn't see any differences between exposure and the control groups. As you can see, this is the bar to show for each clusters, the exposure or the control infants, the numbers are pretty similar. This is a different way to show that controls and the exposure group, they don't have difference when we use the 3 clusters to define them. So both of them goes to these 3 clusters very evenly. However, if we check the nasal microbiome, we found it's very interesting here. As you can see, there are a lot of potentially pathogenic bacteria in the exposure group in the nasal microbiome. So, which means that although there are no like community level disturb as in the gut microbiome, but the nasal microbiome, they have some small pathogenic bacteria colonized in the exposure group. But this may not be related to the prenatal exposure because this may be because of environmental exposure after birth. So like in the 2019, which is still close to the Hurricane Maria, the environmental is different from 2020 when we have the the no. So the environment in the 2018 after the Hurricane Maria may be very different from the environment in the 2019, and the afterbirth. This is the bacteria colonized in the nasal microbiome. So we need more studies, and we are looking forward to some collaborations with the Department of Microbiology to check whether this is because of the environmental exposure, indoor or outdoor. And this is a different way to show all this bacteria which is enriched in the exposed group. The red bar are the exposure group, and the black bar are the control group. You can see most of the significantly different bacteria are increased in the exposed group, not in the control group. So then we ask, because the mothers who were exposed to Hurricane Maria during pregnancy, they have different experience. But we have the questionnaire to ask their experience. As you can see, all of them have experienced the loss of electricity and exposed to molds. However, they have some differences in the experience. And we are very interested to see whether there are some impact on the poor access to food after Hurricane Maria. So we have two groups. We have the mothers who claim they have very poor access to food during the aftermath of Hurricane Maria for at least 1 month. And we also have a normal group. The mother claimed they didn't have any impact or any difficulties to access to food after Hurricane Maria. So as you can see, this is two groups, and we plotted all the relative abundance of the bacteria general in the infant's gut. This is for the gut microbiome. And we identified, this is using the machine learning methods, we found Veillonella is a bacteria which can be a good one to predict whether this mother has been experiencing this prenatal food insecurity or not. So just to use one of the bacteria, you can really tell whether this is infant born to a mother who experienced food insecurity or not. We use different methods to check, and the results are very similar. As you can see, we have 8 mothers. It's a very small number. 8 mothers claim they didn't have enough food. And we have normal access to food. In this group, we have 21 mothers. The infants born to the normal mothers, they have very normal Veillonella. However, if the infants were born to mothers who have full access to food, you can see here, they have less reduced abundance of Veillonella in the gut microbiome. Then we are thinking whether this is because of the breast milk, maybe because the mother during the pregnancy, because of the nutrition, their relative abundance of Veillonella was decreased. Therefore, when they do the breast milk feeding, the infant's Veillonella relative abundance has also decreased. So we grouped the infants into 2. One is breast milk or mixed feeding. So they all receive the breast milk. And another group are exclusive formula. As you can see, in both groups, we've seen the same trend. If the mother experienced food insecurity, the infants have less Veillonella, no matter its feeding method. So our hypothesis is there are some environment, like the gut environmental change during the fetal stage, which caused the colon couldn't accept more of the Veillonella. So, but we need more studies to show that. So this is not only like you can do the probiotics to increase the Veillonella abundance. We need to check whether this is because the gut environmental change. Then we are thinking because the prenatal food insecurity is a top stressor. So whether the difference of the Veillonella is caused by the stress of the mother, not because of the food. So we use another two stressors here. One is maternal access to drinking water, and another one is displacement. So if we divide the mothers into two groups using their status of drinking water access or displacement after Hurricane Maria, as you can see here, we didn't see any difference of the Veillonella in infants, which means that only stress may not be the reason for the Veillonella reduction. The food, the nutrition must play a role here, either combined with stress or just food alone have this effect. It's very interesting that earlier there is a study published in Science Translational Medicine. They identified that Veillonella is one of the very beneficial bacteria for early infancy. So if the infants have less Veillonella here, if their Veillonella abundance is reduced at 3 months, they, like at year 3, they have increased risks for allergy and asthma. And these differences, if they compare these two groups at 1 year, they found the difference decrease, which means that there is a critical window for Veillonella to train the immune system, which is in the early infancy. So we think this is like a clue to show why this disaster or like food insecurity during disaster may cause a high risk of asthma. However, our study still have like it's still like very in the infancy stage, and we want to raise the attention of the public and the research community to do more research on the reasons why the disaster and food insecurity can cause this reduced abundance of Veillonella. So this is the two critical windows here. The 3 months is, you can see the big difference of the Veillonella. So to summarize the first part, I would say this prenatal exposure to Hurricane Maria may lead to a disturbed gut microbiome in offspring. As you can see, there are two clusters there, mostly are for the exposure group infants. And for the nasal microbiome, we didn't see a disturbed microbiome. However, we saw the potentially pathogenic bacteria harbored in the infants who were exposed to Hurricane Maria. And finally, we found that prenatal maternal food insecurity may be associated with the decreased abundance of Veillonella in the infant gut. So the second part is about the lower airway bacteria and the asthma. I think the time is very tight, so I will very briefly talk about this so we can have some minutes to have a discussion. So this picture shows that why the asthma microbiome are very different in the asthma patients, the airway microbiome. As you can see, there are a lot of environmental factors can influence the airway microbiome. And also for the asthma patients, if they have the poor clearance and they have the ciliary damage, this may also cause the environmental change in the airway, which can favor or inhibit some bacterial growth. And in the airway, there are a lot of immune functions, and this bacteria may cause the lung inflammation and cause the asthma exacerbation. And asthma exacerbation can also like cause the temperature change and nutrients change in the airway. And these factors can further influence the airway microbiome. Therefore, it is well documented that the airway microbiome are very different in asthma patients compared to the healthy individuals. However, the problem in the asthma airway microbiome studies are we have identified a lot of bacteria, and some are not like well repeated in different human studies. So we want to know why this is happened. So as you know, asthma is really gender related, as you can see in at different age groups, the gender have a very different asthma prevalence. So there are a lot of mechanisms underlying these gender differences in asthma. However, there's no study really talk about microbiome in this. So we are thinking whether the male and females may use different microbial pathways for the asthma development and asthma exacerbations. Therefore, we want to divide the participants into male and female and do the microbiome studies to see the gender effects in these associations. So this is a human microbiome study which showed that the airway microbiome are different in men and women, as you can see here. This is the green dots are the airway microbiome. Men here has more diverse microbiome than women. And this figure also showed that they observed more different bacterial species within the male and female. So we recruited asthma patients and the control groups in the St. Louis area. This is to plot all the bacterial phylum. As you can see, we have 23 asthma patients and 24 non-asthma controls. And we compared the diversity of the female and male. It's very similar to the human microbiome project. As you can see here, male have higher diversity than the female in the microbiome. And this is the same trend, no matter this is in the asthma patients or in the healthy controls. They have the same trend. Men always have more diverse microbes in the airway than the female. And if we plot their composition of the microbial community, they are very different. As you can see, blue are males and pink are females. We identified there are different bacterial species within the females and males, but very interestingly, we identified four bacterial species. It presents at all the samples, no matter male or female, asthma or non-asthma. However, the difference is like the most abundant one is Streptococcus salivarius. The difference is female has more of salivarius and male has less. However, if you focus on one gender, you can see no matter which gender, asthmatics always have less of this salivarius. Seems like the salivarius may have some protective role in the asthma exacerbation or asthma development. And this is a different way to show, like, as you can see, female in the salivarius high group, females are more in the normal group. Males are also more go to the normal group. But if you have reduced salivarius in these two groups, females are more in the asthma. Males are also more in the asthma. However, you need to use a different salivarius standards because in general, males have less of the salivarius. So you need to have a gender-specific standard to differentiate salivarius high or salivarius low group. So salivarius has been shown that they are beneficial. They can inhibit a lot of biofilm development for the pathogenic bacteria. So this is an in vitro study. This is a good evidence to show it may play a beneficial role in asthma. And in another study, they used azithromycin treatment for the patients with asthma. They found that after 5 months of treatment, these salivarius strains increased for about 5 times. So most of the bacteria are decreased, but the salivarius are increased. So this is another like indirect evidence to show that salivarius may benefit the asthma control. So this is a study we want to identify the bacteria associated with asthma. So the x-axis are the association in females, and the y-axis is the association in males. So if a bacteria is associated with asthma in both female and male, it will be in this item here. So as you can see, we found one of the bacteria, lactobacillus. But interestingly, we found one of the bacteria, Haemophilus. This is only associated with male, but not female. So you can see this too. For the lactobacillus, both female and male, asthma group have higher abundance. For the Haemophilus, female, almost the same, but the male have more Haemophilus in the asthma group. So it seems like the Haemophilus may induce some asthma exacerbation or development specifically for males. There was a previous study showing the same results. They found in the Haemophilus high group, most of the participants, most of the asthma patients are male. So we are thinking there are some like gender specific architectures underlying this asthma. And if we do more studies using some specific gender context, we will feel like yield more findings, and these findings may be more consistent and more accurate. And this is a study we show that the gender effects in association. And the next step for this direction is we want to do some more studies to identify the function of salivarius, lactobacillus, and Haemophilus on the asthma exacerbations, like how these three bacteria in the airway may interact with the immune systems and to cause the type 2 immune fingerprints. So that way we will really tell this is the function of this bacteria and identify the causal role of this associated bacteria. So with this, I want to acknowledge my lab members and my collaborators, especially my collaborators in the San Juan City Hospital. And I'm also recruiting research technicians and postdocs in my lab. If you're interested in the microbiome studies, welcome to contact me, and hope to have more opportunities to share with my future studies. Thank you very much.
Thank you very much, Dr. Wang, for a great presentation. Can you hear me?
Yes.
Yes.
Yes, great. If you have, for the audience, if you have any questions, please feel free to unmute yourself and ask it or through the chat and I will read it. And right now we have one question from Jasenia, I think. Yeah, she says, excellent presentation, Dr. Wang. Would you expect asthma treatment, use of inhalers and or tablets to affect the patient's microbiota and induce different effects?
Yes, I think it's a great question. Yes, I think so. So a lot of asthma treatment and even just the asthma itself can cause the bacterial change. Therefore, when we do the association studies, I think to understand their role is very important because it's very possible these bacteria are a cause or a result of the asthma or asthma medication, but not a causal factors of asthma exacerbation. So right now, all the bacteria, they are either the causal factor or the resulting factors. We need to differentiate them. This is the next step.
Thank you, Dr. Wang. And I have a question. Have you considered also evaluating other microbes such as the virome and the microbiome in the nasal microbiome?
Yeah, thank you for asking. Yeah, my lab is doing research on the sputum virome right now. The data is still like very like not mature enough to share, but I hope I can share in the near future.
That's right. Thank you. Is there another question from the audience?
Mm-hmm.
Yes, I raised a hand. I have a question.
Yes, go ahead, Dr. Banerjee.
This is Deepak Banerjee from biochemistry. Well, I enjoyed your presentation. Apparently, it looks like you're trying to develop the asthma is a microbial disease and probably can be treated with antibiotic in the future. But I have just to make one comment, and then I have a question for you. Almost about 40 years ago, we studied bronchial asthma in guinea pig models, and basically there was a structural changes and an upper area. I'm talking about the tracheal muscle, and there's a changes in ganglioside content, which gives it a high level of pentasialoganglioside, which basically binds calcium and causes the bronchoconstriction. So based on that study, what we found it out on how your study with the microbial system could influence and bring the structural changes on the upper airways, and so they can be very sensitive to immunogen, like there was a histamine challenge or something like that. Sorry, I can't hear you.
Yeah,
we're gonna hear.
Hope I can see the audience.
Okay, yeah.
It's
now.
still facing my slide. Oh yeah, now I can see.
Did you get the question, or should I repeat it?
Can you repeat your question? I think that's the
well,
the
my question is almost as saying that the studies what I did and published in Science, almost 40 years ago, 1982. When you looked at the, it's a guinea pig model because the guinea pig population has about 15 to 20%. They're asthmatics, equivalent to. So if you give them a histamine challenge, you can see bronchoconstrictions. The lung is fine, but the upper ways, which is trachea. So when you correlated that electrophysiological finding with the biochemical finding, we found that there is a very high level of pentasialoganglioside, which is, which is one of the cell surface macromolecules. And the salicylic acid binds calcium. So that was our conclusion was that because of the high level of pentasialoganglioside, it can bind calcium and causes bronchoconstrictions during the influence of allergen. So my question to you, what are the microbial changes you're finding and reflecting to bronchial asthma? How does it relate to the changes, so the structural changes on the bronchial epithelia?
The question is similar to the previous question. Is we really couldn't say the identified bacteria right now cause asthma. It is possibly this is results of the structural change of the upper airway. If that's the case, I think this bacteria can be a good biomarkers, but it will not be a functional role for the asthma. Yeah, that's what I'm thinking, and that's why I feel like to really go ahead to test the function is important. Like in the mouse model or in the guinea pig model, you need to colonize this specific bacteria and check the difference of the immune responses. So I think only using this way we can say whether this is a result or this is a causal factors.
Or you mean to say that the other way, that because of the changes, then the bacteria can colonize. That's like an indirect effect, and that's why you might find some bacteria. It's not necessarily the bacteria is causing the disease.
Sure, yeah. I'm sure there are a lot of bacteria like this, because not only for asthma, like for COPD, for lung cancers, we see this altered airway microbiome. And actually, I found some like similarities between asthma and the COPD because they are all airway obstructive diseases. This bacteria may not really function at the immune pathways, but this is just an indicator of the airway structure change.
Okay. Thank you.
Thank you, Doctor. There's another question from Dr. Jorge Duconge, and he asked, how does the extensive use of sanitizers or the use of masks during pandemic shape the microbiomes and ultimately affect the risk of asthma?
So I will say from the current studies, although we thought asthma patients are highly vulnerable, but the current literature shows that the asthma like emergency visits are dropping a lot during this pandemic. I think the masks really and the social distancing help the asthma population to get infected by a lot of different allergens and pathogens. So, but this is a very interesting question. I'm also thinking whether this social distancing and this hygiene methods and masks can really change the microbiome. I found several studies are talking about the airway microbiome. Even my lab are doing the COVID-19 samples, but we haven't had mature results to share at this moment. But I would say people are really very exciting to see whether the airway microbiome can be an indicator of the COVID-19 severity. So, yeah.
Great, thank you. Dr. Stephanie Dorta Estremera also has a question. Have you begun to study the relationship between the microbial communities linked to asthma and immunological changes, such as T cell and antibody responses?
So the first step is really to identify the bacteria, because the current literature really gives us a very complicated picture of the related bacteria. So one of my lab we want to do is really to best use the human population to identify the bacteria and then do the immune functional studies. And we are collaborating with the lab, and we are using, it's very exciting to use the humanized mouse model, which means the human immune system has been engrafted into the mice. So we really can see the interactions between the bacteria and the human system in the mouse.
Okay, thank you. And Eduardo Lemuertosado also asked, since the relationship of asthma development and exposure to fungal spores have been well documented, there are any studies focusing in the microbiome of these asthma patients?
Asthma patients, you mean my asthma patients or?
Just in general.
Okay, yeah. So there are studies starting on the fungi and the asthma. This is like, I think it's unavoided area because fungi, there are very established relationship of fungi and asthma. So although my lab haven't started to tackle the fungi part, I'm very interested and very exciting to do collaboration on this, because you know that the microorganisms are in a row, in a whole, right? You couldn't just do one part. You really need to see their interactions and their interactions with humans.
Right. That's great. Thank you, Dr. Wang. If there are no additional questions, we conclude today's seminar. Thank you so much for being here today with us and for an excellent presentation.
Definitely. Thank you very much, Dr. Wang. I joined Lorraine's work. Thank you for your time with us.